All right, this is going to be the first of 4 short videos regarding cell communication, or sometimes called cell signaling. And in this first one, we're going to talk a little overview about what cell communication is. And then we're going to talk about the signal aspect of cell signaling, of cell communication. And what I'd like you to be able to do after you're done watching this is I want you to be able to explain the concept of genes and proteins being conserved. And so this is an evolutionary concept. I want you to be able to define the 4 components of cell communication. And I want you to compare and contrast the different types of signals based on the distance that that signal has traveled from the source to the receptor. So what is cell signaling? Well, what cell signaling is, it is the way that cells interact with their environment and how they can communicate between one another within a multicellular organism, as we see here in this impala, or even single-celled organisms like bacteria and yeast even communicate to one another. So if we take a look at this, this will show us the entire breakdown of cell signaling. So let's take a look. So the impala is being chased by a cheetah, and the impala senses a cheetah. The impala's brain signals to the adrenal glands to release a signal called epinephrine. Now that epinephrine gets released into the blood and eventually reaches the various cells in the body of that impala, specifically, in our example here, the muscle cells. Okay, so in the leg of the impala, the muscle cells, because we know these muscles want to start contracting like crazy so that impala can get away from the cheetah. Now, this constant contraction of muscle cells requires a very fast source of energy. Glucose happens to be a very good source of energy, and the cells get this glucose from stored glycogen, which are molecules that are just glucose molecules stuck to one another. But to break those glucose molecules off of glycogen, they need an enzyme. So this enzyme catalyzes the reaction of breaking glycogen down into glucose. Now, we don't want this enzyme to be always active. We want it to be off sometimes because, right, the impala doesn't necessarily need all this glucose all the time. But when it's being chased by a cheetah, it definitely wants this reaction to occur, meaning it definitely wants this enzyme to be turned on. So how is that decision made? Well, as we said, the brain sends a signal to the adrenal glands. The adrenal glands releases epinephrine to the blood. Eventually that epinephrine, that molecule, binds to a receptor on the surface, embedded within the membrane of muscle cells. When the epinephrine binds to that receptor, the receptor changes in conformation. That change in conformation turns on another protein, which then turns on another protein, which then turns on another protein, and this process is called signal transduction. Finally, the last thing to get turned on is this enzyme here that breaks glycogen down into glucose. That is the cellular response, breaking glycogen down into glucose so that the impala's muscle cells will have plenty of energy to work with and power the contraction of those muscle cells so that impala can get away from the cheetah. So what we have here then is the signal, that's epinephrine, the receptor, that's this molecule here, the signal transduction pathway, okay, that's the steps that connects the activated receptor to the cellular response. And then we have here the cellular response. Okay, so we're going to go through in the next 4 videos each one of these, and what we're going to start with then is this signal. But first I want to make sure that we all understand, and I want to stress one more time, is that cell communication is essential not only for unicellular organisms, right, like bacteria and yeast. It's important for mating between 2 single-celled organisms. It's involved in something called quorum sensing, a way that single-celled organisms have a way to measure just how densely populated a particular environment is. And then in multicellular organisms, the pancreas cells release insulin that go on to tell other cells to take up glucose from the blood. What we just saw, the example we just saw, the adrenal gland cells release epinephrine, and that signals to muscle cells to start breaking down glycogen. So there's all sorts of examples of where cell communication is absolutely essential. Now, the proteins involved in cell communication are encoded by genes that are very highly conserved. And what this means is these genes have evolved a billion years ago, many, many years ago. And these genes have been, as organisms speciate, as they evolve, they carry these genes with them, and they're so important that they are always being selected for. And everything from bacteria to humans use these same sets of genes that are involved in cell communication. So it's a very highly conserved process. Okay, the signal. So when we talk about the signal, what we mean is right here, right? That molecule that has been either released into the blood or released into the matrix, the intercellular matrix. But that is what is eventually going to bind to the receptor. But we're going to look at the receptors in the next video. For right now, let's pay attention to the signals. And we're going to classify the signals based on just how far away the source is from the receptor. So local signaling versus long distance signaling. Now, the most local signaling there is for the two cells, the cell that is the source of the signal, the cell that is reciprocal, or the receiving the signal, to be actually connected to one another and have holes in them that they actually share cytoplasm. So, for example, if we're looking at animal cells, gap junctions. Okay, we talked about gap junctions in chapter 6, and those allow cytoplasm and therefore the signals themselves to migrate into the other cell. Plant cells have something called plasmodesmata, but same idea. Holes within the cell wall, holes within the membrane, through which molecules can directly pass. That's the most local signaling there is, these cell junctions. Now, if we bring it out a little bit, what we end up with is cell-cell recognition. So now we've got our two cells, right? The source of the signal here on the left and the receiver on the right. But now they're not sharing cytoplasm. There is no holes in the membranes through which these signals can pass. But the signal, instead of being a free-floating object, is directly connected to the source cell. These are like those glycoproteins that we had discussed in chapter 6. Things like blood type proteins. They don't leave the cell. They are bound to it. Now the receptors here, they're still the same sort of receptors that we'll look at before. But the idea is these receptors then, this cell, the receiving cell, has to actually bump into the target cell so that it, quote, feels its signal. And once the receptor bumps into one of these molecules that's bound to the surface of the target cell, or of the source cell, that changes the conformation of that receptor, and then we get a signal transduction pathway going that we will learn about in the third video. Then if we now come a little bit farther out, we have something called paracrine signaling. Now, the target cell, the source cell releases free-floating signal into the space between cells. And the target cells have the receptors that will receive that signal. So we have this, but the difference here is that the cells still have to be relatively close to the source. Okay, they can't be super far away, probably 3, 4, 5 cells max. We can take paracrine signaling and we can use an example, a specific example of the neurons. So here we have a electrical signaling that's going to trigger the release of neurotransmitter. The neurotransmitter is going to travel across that space between the two cells, and that's called the synapse, and then the target cell then is going to receive that signal. That's an example. So this neurotransmitter signaling is an example of paracrine signaling. And then we have the most long distance signaling, and that is endocrine or hormone signaling, where the source of the signal is quite far away from the target. And so what happens is that the source of the signal, the endocrine cell, maybe we're talking about a pancreas cell releasing insulin, or an adrenal gland cell releasing epinephrine, but that cell is going to release the signal into the bloodstream, and then it'll travel through the bloodstream, it'll bump into all of the cells, and any cell that has a receptor that specifically receives that signal will then respond to it. And so that's the longest long distance signaling there. So again, make sure that you can go back and review those slides and that you can explain the concept of genes and proteins being conserved, right? Again, that's that evolutionary term. Define the 4 components of cell communication, and then compare and contrast the different types of signals based on the distance that they've traveled between the source of that signal and the final target.